Hello friends, today we are going to discuss about one of the important topic about bioinformatics. It's biological databases, tools with special reference to molecular docking. Bioinformatics. Bioinformatics. It's databases and tools with special reference to molecular docking. The word bioinformatics is a combination of two terms, the biology and the information technology platform. What exactly it means? The biological database, which act as heart, and the IT, that means it acts as a brain, and these two are interlinked through a set of tools, that means both basic and essentials, so that we can manage, analyze, and and what we call MAM principle: manage, analyze, and manipulate. The crux point of bioinformatics based on this MAM principle. That means we can manage, analyze, and manipulate varied or a mammoth biological information pertaining to different resources. Why would like to do all these things? Why would like to manipulate the information in the databases? Because it is essential to analyze, it is essential to manage, to understand or construe varied biological processes like gene expression, or to discover a novel drug, or even for three-dimensional structure of a protein. To understand these things, it is essential to do what we call MAM principle: manage, analyze, and manipulation of biological data present in the IT platform, what we call databases. For example, what exactly? Let me take a simple example. A scientist have sequenced a particular DNA sample and upload the same in a database, what we call IT platform. And another scientist doing the same research may utilize the same for his or her research according to their research plan. And again, that means the modification of the same will result in a different result. And they may upload their modified version. That means in first context, they are giving some X information, the first scientist. In the second context, a Y information has been added. What we call collection of biological information and manipulation of the same so as to understand or construing of varied biological process. It's about the whole dimension or the crux point of bioinformatics. I told you already, MAM principle. So to understand varied biological process, we have to follow these principles, what we call MAM. But the thing is, whatever we say, it is completely a prediction. Because what in silico analysis, it is not a wet lab just like biotechnology, this is dry lab. Just like with the help of some software tools or powerful algorithms, we may predict the possible outcomes, but it should be corroborated with the wet lab, then there will be a meaningful result. So it's about the bioinformatics. Okay, the word it implied, biology and IT platform, that means biological databases. This is the essential component. Say it is a heart and IT is a brain, and they interlinked so that based on MAM principle, what we call manage, analyze, and manipulate through different tools and techniques. Different tools, what we call algorithms, for instance, say software programs. Okay, it's about bioinformatics, and bioinformatics is actually a multidisciplinary subject. Consider it is as bioinformatics. There are several sub-disciplines connected to bioinformatics. Say functional genomics. I'm mentioning some functional genomics, then structural genomics, evolutionary biology, drug design, genome annotation, proteomics, DNA chips or microarray, No, these are different subjects that connects to bioinformatics. I have already discussed. It's a multi-dimensional subject. Its application base is very wide. Say, functional genomics. The word it implied identification of the gene and its functional aspects. That means we knew that genome is nothing but the entire DNA content of a cell or an organism in genomics. So again, there are functional genomics and structural genomics. Broadly, the word structural genomics implied that three-dimensional structure of a protein. Okay, then drug designing. No, we are in the era of radicalization, what we call revolutionization of new novel viral strains. So it is essential to discover drug against the same. Say, identifying the active site of the concerned antigen and neutralizing the same is very important. It's what we call drug design. And evolutionary biology is the what we call molecular phylogeny. Molecular phylogenetics, one of the main branch of phylogeny. We can analyze different hereditary molecules so as to understand the evolutionary relationship of an organism and construct a phylogenetic tree. Then proteomics, the word it implies about a large scale protein structure and function analysis. Now we knew that proteome means the whole complete set of a protein. Okay, that's like genomics. It's the opposite domain. Genome annotation. The word annotation means linking or attaching the biological information to genomic elements. See, here annotation means that kinds of three types. It's like first you have to identify the non-protein, that means non-coding portions of a genome. Then what we call second step is gene prediction. That means identifying the possible genomic elements and attach or link their biological information to the top of the genomic elements. This genome annotation is also one of the main branch, that means interlinking issue of bioinformatics. And DNA microarray. It's like microarray. I mean chips what we call. No, these are all broadly useful in identifying different levels of gene expression in a tissue, even disease. So these are all different connected issues. That means how different branches use bioinformatics, because it is a basic fundamental aspect for in silico analysis. So with the help of software and even powerful algorithms, we can predict the possible outcomes. Say for example, drug design. Say for example, comparison of different sequences. Say a nucleotide sequence with that of the database, so that we can ascertain whether our strain, that means our sequence, is a novel or already reported. That means it's about different applicational role of bioinformatics and how it broaden the basket of bioinformatics. Okay. Then, as we have discussed, the databases. We have already discussed that databases is the central theme of bioinformatics. Without database, we cannot collect. It's like entire globe has been connected through the biological database, of course through IT platform. And these biological databases addition, management, manipulations, and again upgradation is all regular affair of bioinformatics scientists. So understanding databases is very important here. And so we will discuss some of the important databases here. Some of the important databases. Say the databases can be categorically divided based on the kind of biological element involved. For example, say nucleotide sequences. Nucleotide sequence database. Mostly I have discussed about NCBI and its particular wing is called GenBank, EMBL, PDBJ. Then the protein sequence database. Protein sequence database. Swiss Prot. Protein sequence motif database. Motif database. ProCite. Then macromolecular database. Macromolecular database. PDB. We got protein data bank. Protein data bank. Okay. Just I'm discussing some of the important regularly popularized databases. Okay. say this is called nucleotide sequence databases. We can say these as primary nucleotide sequence databases. Say NCBI, National Center for Biotechnology Information. National Center for Biotechnology Information. It is a US based database established in 1988 as a branch or as an offshoot wing of National Laboratory for Medicine (NLM). It is at the institute what we call NIH, National Institute of Health, and this NCBI's primary wing is called GenBank. The word it implied, it's a DNA nucleotide sequence database. Since 1992, it is operating this GenBank, and this GenBank is very essential because in addition to the other databases and even individual molecular laboratories, say for example EMBL, European Molecular Biology Laboratory, it's one of the important database, and DDBJ, DNA Data Bank of Japan, DNA Data Bank of Japan. This is an European Molecular Biology Laboratory. These are all primary nucleotide databases. Okay. Then there is another wing is what we call Unigen. Here we can get the information with respect to gene clusters, that means DNA clusters and representative genes. That means the nucleotide sequence information of GenBank can be available in the form of clusters and representative genes. So these are all about different nucleotide sequence databases. NCBI, National Center for Biotechnology Information, and this is the main wing is called GenBank, and it is what we call primary nucleotide sequence database. Since 1992 is operating, and it is very essential because it coordinates with individual laboratories and important primary nucleotide sequence databases like EMBL, European Molecular Biology Laboratory, and DDBJ, DNA Data Bank of Japan. Okay, and Unigen is one of the, it's like a subsidiary wing of GenBank. These are all, that means NCBI, GenBank, Unigen comes under GenBank, and Unigen comes under NCBI only. We have also genome biology. We can get the information with respect to genome of different organisms. So these are all, that means the GenBank, the Unigen, genome biology comes under NCBI head. Okay, then this is about nucleotide sequence database of GenBank in clusters and representative genes in Unigen. Then regarding protein sequence database, we have important database called SwissProt. And regarding protein sequence motif database, one more database called ProCyte. And regarding macromolecular database, it is called PDB. This is very important. PDP, Protein Data Bank, is a crystallographic data bank because the three-dimensional structures of varied macromolecules like DNA and RNA are available here. 90% of the 3D structures we can get in PDP are based on X-ray crystallography. Apart from X-ray crystallography, the NMR spectroscopy aided structures we can get in PDP. It's all about like retrieving the information and manage and manipulating according to our source, and again uploading in an upgraded version. In PDP, NCBI, these are what I have already discussed. Manage, analyze, and manipulate. You retrieve an information, you manipulate it according to your things, and that means what we upgrade, then again upload. Some other scholar or scientist may use the same, then the UME continues like cyclical phenomenon. Just you retrieve, then you use according to, that means manipulate, then upgrade, then you upload. So these are all create a wide set of database, what we call a mammoth database, for understanding various biological phenomena already discussed. we can understand the gene expression, we can understand the protein motif structure, we can understand a proper drug design, it will be novel drug designing which is essential against a wide area of infectious biological diseases. There are a lot of different, that means this is how the databases serve the purpose. It's like information pool, a resource, so that understanding the resource and manipulating according to our needs is a very essential step in proper utilization of bioinformatics. And there are different databases, okay? NC by Gen by EM by LD by Swissplot, ProSite and PDB. Okay. Regarding tools, there are about different databases. We already discussed about the second vital element. How we can use the database is very important. To understand the tools, we have to understand the working mechanism of bioinformatics. Bioinformatics based on three essential components. Number one, creation of database, creation of biological database. Number two, algorithms and statistics. Number three, analyze and interpret. So that means we have discussed about different biological databases. And before we go into the tools, we have to understand the working, how the bioinformatics generally actually works. Say, first we have to create a database. What is a database? That means with respect to biological database, what exactly it is? It is nothing but collection of biological information on a computer, manipulation of the same so as to create a varying domain or subset. Collection of biological information. Collection of biological information. That leads to creation of a varied or a different subset. We have all discussed, you can retrieve and manipulate according to our source, then upgrade. That means creating a novel version, a new version of a particular biological element is a basis, is called database. Okay, and it contains a mammoth of biological information. Then after we have created a database. How can we use it? This is very important. These are all interlinked, How can we use, say for example, I have retrieved a particular sequence and I would like to compare with another. That means, for example, I have a particular DNA sequence. I would like to ascertain whether my species is a new one, or my strain is a new one, or already reported one. How we have to proceed? First, we have to compare my sequence with that of the existing databases. Then it will give a result whether it is a new one or a reported one. How we can do? There are tools, what we call tools. These are called functional issues. What means to analyze, interpret, we require some tools, what we call bioinformatic tools, simply called algorithm. This is software programs. Algorithm is nothing but, algorithm is a procedure or a formula to solve a problem. And software is nothing but a set of instructions that directs a computer to get a thing done or to undertake a specific task. Say it's like a sequence of steps we are directing which can direct a computer to get the things done or to perform a specific task is a software. And algorithms will reduce the errors in the software. That means algorithm enhances the efficacy of software, and this efficacy of software is essential in analyzing and interpreting the results. If we apply a poor software, we cannot get appropriate results. So powerful techniques, that means powerful tools and advanced software versions is very, very important. For example, a modeler software version 9.17, it is very important for homology modeling of a particular protein. Or say iGemDoc version 2.1 is essential or important molecular docking tool. That means this algorithm based software is essential in identifying, that means in analyzing our data, whether we get the new one or the old one, or for three-dimensional structure. So it's about how the things done, the information has to be processed with the help of algorithms and advanced statistics, simply called software programs. And we can so ultimately that means the combination of 1 and 2 leads to proper analysis and interpretation. If you have a powerful software, that means the appropriate novel software that analyze different data sets, we can get that means we can properly interpret, and that gives a biologically meaningful information. So collection of database, algorithm, that means creation of using of appropriate tools, particular algorithms, so that we can analyze and interpret. These are the three essential domains of bioinformatics. Then we have discussed about databases and how the working mechanism, and the help of tools we can get appropriate interpretation. Okay. Then regarding tools, now the most important tools are BLAST, FASTA, emboss. Here for BLAST, first we will discuss these two important things. Basic local alignment search tool. See, BLAST P, BLAST N. No, BLAST, basic local alignment search tool. Though there are different tools, and this is very, very important. Without BLAST, you cannot expect any credible outcome. Okay. Then here, the word implied basic local alignment search tool. What exactly it will do? For example, we have discussed with the example. I have got a nucleotide sequence. I would like to compare with the existing sequence, whether it is a new one or the reported one. That means comparison of biological sequences is the main duty or theme of BLAST. This BLAST software program will compare the query sequence with that of the databases. The query sequence might be a nucleotide sequence or a protein sequence. That means, for example, if it is using protein sequence, then we can say BLAST P. Then if it is nucleotide sequence, if it is called BLAST N. So depending on the kind of sequence, we can get BLAST P and BLAST N. This is very important. Then FASTA. Here FASTA here means fast all. Fast all. It's like ubiquitous in bioinformatics. Any word, any alphabet we can use. For example, if it is using, that means if it is using with respect to nucleotide, we can say fast N. If it is protein, fast P. But only thing is, it is an alignment tool. It's an alignment tool. Alignment tool. If we are using nucleotides alignment, you can fast N, fast P. Then regarding other, these are very important tools. Okay. European biology, molecular biology, open software suite. Another software, European molecular biology open software suite. Here, in examination point of view, they might ask you even the abbreviations also. What is emboss? European molecular biology open software suite. Then RASMOL. You can have other things. Say RASMOL. Prospect. Prospect. Copia. Consensus pattern identification and analysis. COPIA stands for, RASMOL, prospect and COPIA. RASMOL is one of the important tool. That means prospect COPIA also. RASMOL is one of the important tool for image. That means for displaying the structures of DNA. Displaying the structures. This is very important. Displaying the structures. And prospects. Prospect means probability of structural, that means a prediction. That means prediction of protein structures. is the main objective of prospect. Prediction of protein structures. Copia is useful for protein motifs analysis. Protein motifs. That means depending on the kind of objective, there are different softwares. Copia, consensus pattern identification and analysis, for particularly useful for protein motifs analysis. When prospectus, the word implied, it's like prediction of the protein structures. It's all about probabilities. That's it. I already discussed. It's about dry lab predictions. Okay. Then RASMOL, it is useful for displaying the structures of DNA, RNA, and other molecules. So displaying domain for RASMOL, prospect for protein structures, and copia for protein motifs. Emboss and FASTA for an alignment. BLAST for it's like comparison. BLAST is for comparison of biological elements. These are all about different techniques, the essential tools used to say BLAST, FASTA, Emboss, RASMOL, Prospect, and Copia. Apart from these, there are also different tools based on the requirement we can use accordingly. Okay. Next, the most important one, the molecular docking. This is one of the object, molecular docking. Molecular docking. Docking literally means best pose. Molecular docking literally means best pose. It means we have already discussed about drug design. One of the interdisciplinary, since bioinformatics is a multidisciplinary theme, it also includes different wider applications. Say drug design is one. How we can discover a drug against a particular infection. Say for example, infection is because of a protein, say antigen, which is nothing but a protein, for example. This is assume this is as a protein, okay? And this is called active site, this particular portion. This is the whole molecule, this is the active site. Neutralizing the active site is very important. How we can neutralize? By inserting appropriate molecule into so that the target site become deactivated. Here the thing is, for example, here proper orientation of the drug. That means conformation of the drug is in such a way that its orientation should best fit into the active site. What we already discussed. Best fit. Docking means best fit. Say what exactly I mean to say. Say we would like to predict different conformations when we can ascertain which is best in tune of best fit. This is very important. Say this is active site, already discussed. This is docking. This is drug 1. What we call ligand. This is receptor. Ligand receptor interaction. Okay. Ligand is a drug, not fit. Second one, not fit. Third one, best fit. That means we design different drugs and we can predict based on different orientations. We predict different orientation and decide which one is best. That means the best orientation will best fit. Best orientation will best fit. That leads to strong association, strong linkage. Is very important. Higher the efficacy, that means higher the orientation and higher the association or efficacy, and higher is the possibility of reducing the infection. So this is a crux point for drug design. What exactly I meant to say is, say this is a target protein. This is a target protein. And this is the active site. Okay. We would like to discover a new drug. Different orientations we would like to ascertain so that which orientation is best fit. Say, example, the first orientation, the second orientation, the third orientation. Here ligands, what we call drugs. Strong, stable, complex That means ligand and receptor is important in neutralizing the target element. That means strong association is essential. For that strong bond, that means strong complex, strong stable complex is essential. In turn, based on proper orientation. Proper orientation. Proper orientation is best. For example, this we have tried but didn't get properly because its orientation may not best fit what we call best pose or best fit cannot be done properly because if we can have a stable complex come to the second orientation not proper because its orientation is different but means conformation is first and orient with proper orientation. Then the third conformation, its orientation is good because say for example we can get here. It completely forms a stable complex. The third one, third one is best fit. The third one is best fit. That means this is a drug, for example.